109 biology, chapter 1, introduction to biology. 1.1 says that definitions of biology. Biology came from two Greek words. Bio means life and logos means study. So biology is the scientific study of life or living things. That means biology deals about living things or about life. Under 1.2, why do we study biology? Why we do study biology? We study biology to understand how living things works. How they function. Normally biologists deal about or study about living, so that while we are study biology, how living things work, how they are function, how they interact with their environments or the living things and the environment, how they are interact or how the relationship can be created. So biology touches our everyday life in many ways. For example, biologists have discovered drugs that are used to treat different human diseases. There are so many diseases are there in our world, so biologists discover different drugs to treat different human diseases. Biologists are also working on problems that critically affect our lives. There are problems in our world, so biology try to solve these problems in different ways that affect our lives, such as how our animals and plants body work. Biology or biologists try to solve the problems such as how animals and plants body work. Also how ecosystem work. How advancements in genetics. Normally genetics is the study of the transfer of genes from parents to offspring. So how this should be advanced in order to use for human beings for the futures in different ways. So biology gives us an advancement in genetics. How cell biology transferring to medicines. While we are learning under biology, we learn as a chapter in different class level about cell biology. It's very important. Cell is the basic and the functional unit of life, so that how this cell biology are transferring to medicines or to make medicines and agriculture, or how it involved in agriculture in order to increase the productivity of animal and plant yields. So biology is very important in order to transferring agriculture and the use of forensic biology to investigate crime. Also biology deals as how ecology is helping societies evaluate environmental issues. There are different environmental issues nowadays, so under biology, while we are learning ecology, it helps us as societies or people in our world, including our country. It helps to evaluate and to solve the environmental issues such as global warming. Nowadays, there is a high flood are formed in our world, including our country. So biology helps to solve such problems. The increasement of heat in our world, for example, nowadays that affect our European countries. Now there are so many people that are affected due to the increasement of heat. So biology is involved in this. Because of that, we learn biology due to this. How fermentation is used to produce alcoholic drinks like beer, wine, sauce, and so on. Again, biologists treat patients suffering from AIDS, tuberculosis, and some types of cancers. That means biologists are trying to find different medicines in order to treat patients that are suffering, such as HIV/AIDS or AIDS, tuberculosis, and some types of cancers and other diseases. 1.3 the scientific methods. As a biology, while we teach biology or while we learn biology, we should follow the scientific methods. There are different steps of scientific methods. So biologists are always curious, or they are active about why things happen and how things happen by scientific methods. The general steps of scientific methods are, the first one is observation. Observation can be takes place in two ways, directly or indirectly. Directly by using our sense organs. We have about 5 sense organs. With these sense organs, we are collecting informations that are to be a topic for our researches or for our researches. Or indirectly using scientific tools such as microscope and other tools, we are finding a topic, which is very important for our scientific methods to use in doing any researches. The second step is asking questions. After we are observing, we raise a question, so that asking a question, which is a topic. So the observation usually leads the scientists to ask questions. That means we have got a topic under asking questions that we are observing. The third step is forming hypothesis. Hypothesis means proposed scientific evaluations or possible answers for a set of questions. A scientific hypothesis should be testable. Hypothesis means we propose by evaluating different written materials what will be our answers in the futures, or we are estimating depending on the different researches and the different informations. The fourth step is testing the hypothesis. We are testing the hypothesis means we test it through experiments. As a science and as a biology, any topic should be experimented. It should be experimented. The scientist comes up with whether it supports or rejects the original hypothesis under our testing hypothesis. The fifth step is making conclusion about our findings. After we are tested under experimentation, then we should make conclusions about our findings. So scientists ask whether they could still be right in light of the new information gathered during experiments. That means we collect and we experiment our informations, whether it is true or not. We are comparing with our hypothesis. If our experiment right or correct, we accept it. If not, the scientists reject the hypothesis and try to come up with alternate explanations or new hypotheses. When we complete some work, they write a paper that explains exactly what they did and the result they obtained. Then they submit the paper to scientific journals in their fields. In addition, the findings will be printed in scientific journals as words and assist teachers and students in the fields. That means under the field of biology. Under the next topic is 1.4 tools of biologists. Biologists, they are using tools. These tools can be divided into two as laboratory tools or that we are using in a laboratory and tools that we are used in the fields. So biologists use different types of tools in a laboratory and the fields under 141 laboratory tools of biologists. That means the tools that we are using in laboratories. Maybe the tools that are used in laboratory also they are used in fields, but most biologists tools are used in a laboratory. One or the first tools that we are using in laboratory is hand lens. Hand lens has a higher magnification than our naked eyes. Under biology, while we deal about microorganisms are not seen with our naked eyes, because of that we need a hand lens or a microscope that magnifies more than our naked eyes. So that hand lens is that has higher magnification than our naked eyes. The important function of microscope is magnification and resolution. Magnification means the number of times larger an image. That means one object or specimen should be magnified larger than an image in order to see easily their structures and any necessary materials than the real size of the objects. Resolution can be defined as the ability to distinguish between two separate points. This microscope can be divided into as light microscope and electron microscope. A light microscope that uses a beam of light to view specimens or our objects or our specimen. So a light microscope can be divided again into two as simple light microscope and a compound light microscope. A simple light microscope, which consists of a single lens that contains one lens, whereas a compound light microscope consists of two or more than two glass lenses. Both of them are used in order to magnify small objects, and they are using a beam of light in order to see the objects. The second type of microscope is electron microscope that are using a beam of electrons and electromagnets rather than the beam of light to enlarge the image of an object or to see small things. This microscope provides a higher magnification. That means an electron microscope has a higher magnification than light microscope and are used for observing extremely small microorganisms such as virus. They are very extremely small, so that in order to observe microscopes and their structures, we are using in laboratories, especially in international laboratories, use this electron microscope, which magnifies than light microscope. The next or the third lab tools or laboratory tools is glass slides. Microscope slides are used to support an specimen being examined under the microscope. Glass slides which put our specimen or objective that observed under microscope on it. We call it glass slide, whereas the next Laboratory tools cover slip. Cover slips are used to cover specimens, protect from any chemical or dirt. That means that covers our specimens on slides that observed under microscope, which covers the specimen. Again, protect from any chemical or dirt our specimens. Again, to protect the microscopes also from damaging our microscope. Again, prevent the slide from drying by lacking the moisture and provide better view under the microscope. Another laboratory tools is autoclave. Autoclave is another laboratory tools that used to sterilize or kill microorganisms on different biological samples. Maybe while we are doing experimental laboratory, we have different samples. On these different samples from the air and the others, maybe unwanted microorganisms arrive on our specimen or on our sample, so that this autoclave used to sterilize by using heat to kill microorganisms which is not needed. Another laboratory tools is incubators, which used to maintain a specific environment for culturing microorganisms. To study about microorganisms, we are using microorganisms in our laboratory. In that case, we put our culturing microorganisms in incubators in order to maintain the temperatures of the environment to keep our microorganisms used for studying. Another laboratory tools, petri dish, are another tools which used with gelling culture media or growing a culture media on it, such as by using agar and placed in the autoclave to sterilize. The next one is culture tubes are used to culture microorganisms or grow microorganisms, used with solids or gel with liquid broth culturing media, then placed in autoclave for sterilization or for keeping the microorganisms on it. Another laboratory tool is flasks. Flasks are used for mixing solutions without spilling out of contents, which is very important in order to mixing our mixtures which have different shapes and size. Another one is balance. Balance is another laboratory tool which is useful for making up solutions accurately, weighing specimens. That means used for weighing our specimens in order to make a solution or mixture in laboratory. Another material or laboratory tool is dropper, which is used to require to control the amount of solution being added to a reaction. While we making a reaction, by using these droppers, we control our amount of solution added to reactions. Another one is tongs. Tongs is another laboratory tool which is used to lift or pick up hot objects such as heated crucibles, beakers, dishes, or flasks, and so on. Maybe while we are touching with our hands, maybe damage our hands. Because of that, we are using these tongs while we are pick up objects in laboratory. In addition, touching any laboratory tools with our hands are not advisable because they may be danger for our hands and for our health. Because of that, we are using these tongs. Another one is dissecting tools or toolkit, which used to dissect animals such as frogs. Laboratory, while we are dissecting different animals, we are using this dissecting toolkit, which contains different materials which used for dissecting animals like frogs and so on, to study their internal structures of their body. It consists a catheter like groove, probe, scalpel, surgical scissors like Mayo scissors, dissecting forceps, dissecting pin, and so on. These are included under dissecting tools which involved in dissecting animals to study their internal organ. Another one is dissecting pan, used as a pan on which the specimen kept. After we dissecting animals, we are put on this dissecting pan and study their structures. Another laboratory tool is crucible, made up of ceramic or metal used to melt elements, used to melt our specimen. The next one is beaker. Beaker used for making up solutions, holding hot or cold water or ice baths and so on. This used for making solutions, again holding a hot or cold water or ice bath, which has own numbers. By keeping their numbers and limiting their amounts, we are making a solution on this beaker. Another laboratory tool is hot plate. is useful for heating solutions. A hot plate that used for heating the solutions by releasing heat, making up an agriculture media, and so on. The next one is pH meter is used to measure the pH acidity or basicity of substances, to measure their pH, whether their acidity or basicity, by using this pH method. These all are about our today lesson, students. We complete our today lesson here. We continue next time. Goodbye.